Trillions of microbes live in our lives, lurk in the air or water, the foods we eat, and the objects we touch. Nevertheless, most of us are healthy most of the time, unaware of the constant battle between body and microbes. Even when we have we almost always send them off quickly. Our usual state of good health is due to three lines of defense: external barriers that keep microbes out of the body, non-specific internal defenses that combat all invading microbes, and our immune system, which results specific microbes. The body's first defense against microbes are the skin and the mucous membrane of the digestive and respiratory tract. The dry dead cells, filled with hard protein that cover the outer surface of the skin, prevent microbes from landing on the skin, from obtaining needed water and nutrients, while acids and natural antibiotics secreted by the sweat and sebaceous glands actively inhibit the growth of bacteria and fungi on the skin. The membranes of the digestive and respiratory tracts are protected by mucus, which contains antibacterial enzymes that destroy the cell walls of the bacteria and physically traps microbes that enter the body through the nose or mouth. Cilia on the membrane sip up the mucus, microbes, and all until it's either coughed up or sneezed or swallowed. Swallowed microbes enter the stomach, where they encounter the combination of an extreme acidity and protein digesting enzymes that kill many microbes. Farther along the digestive tract, bacteria, harmless to humans, secrete substances deadly to other invading bacteria or fungi. While these defenses are good, the warm, moist mucous membranes are more vulnerable to invasion than the skin. Disease organisms that enter the body through skin or mucous membranes run into three non-specific internal defenses: standing armies of phagocytic and natural killer cells, the inflammatory response, and fever. The immune system is a collection of cells, tissues, molecules that protect the body from numerous pathogenic microbes and toxins in our environment. This defense against microbes has been divided into two general types of reactions. First is the innate immunity that responds to all kinds of pathogens, whether your body has seen that pathogen before or not. Pathogens are agents which cause disease. Second is the adaptive or acquired, which develops more slowly and requires your body to know information. Every animal has innate immunity, even sponges, but only vertebrates have the adaptive or acquired kind. You were born with an innate immune system, and from the moment you wiggled yourself out of the scary world, it has been protecting you. The thing about it is that it doesn't care what it's killing, whether it's a virus, a fungus, or a bacteria. Its job is just to keep. The first line of defense in keeping foreign objects out are the skin and the mucous membrane. The skin is the fabulous bag for your body. It's stretchy and waterproof. It covers your body and keeping organisms in. It protects us from unwanted substances from getting in. We've also got mucous membrane, which provides us barrier from microbes trying to sneak in. It lines all of the internal surfaces that are exposed to the outside, like the lungs, the inside of your nose, and as well as some other parts of your body. It produces mucus, that is a viscous fluid. Second line of defense is your inflammatory response. It has specialized cells in your connective tissue called mast cells that constantly search for suspicious objects, which is usually unknown proteins, and they'll release signaling molecules like histamine. Histamine makes your blood vessels more permeable, which allows fluids to flow to the affected area, and that's what causes inflammation. But it also brings a ton of white blood cells, infection fighters, to go all power on whatever is trying to make in. But sometimes, something gets into you that is not actually dangerous, like a pollen, a dust, or a peanut. If your immune system triggers inflammatory responses anyway, even though it's not a big deal. This is what we call allergic reaction. And you know what it's like with the swelling, the redness, the mucus production, and the itching, and occasionally a little bit of death. That is why we take antihistamines to suppress the lymphatic system, aids the circulatory system in transporting fluids, and is a vital part of the immune system. In and around the cells of the body, extracellular fluid collects waste products and carries nutrients and oxygen to the cells. Some of the fluid travels through the capillary. Some, especially if they contain proteins too large to enter the capillary membrane, circulate to the lymphatic vessels. Lymphatic vessels flow through several lymph nodes. These lymph nodes contain concentration of white blood cells, which kill bacteria and other harmful agents trapped in the node. The tissue fluid. The acquired system keeps an eye after any foreign substance, toxin, or virus, even parts of those things that can be a telltale sign of an invader. We call those signs antigens, a word that comes from antibody generated. An antigen is anything that causes your immune system to ID a pathogen and then they aren't cells, but highly specialized proteins produced by these cells to recognize and help kill intruders. They can't kill invaders themselves. They serve as scouts, attaching themselves to the bad guys and then releasing chemical signals to nearby phagocytes, alerting them that it's dinner time. The acquired system has to learn as much as it can.
Production of lymphocytes. Lymphocytes are a critical part of the human immune system. There are two types of lymphocytes, T cells and B cells. Around 85% of lymphocytes seen in the blood are T cells, also known as T lymphocytes. T lymphocytes are responsible for cellular immunity. The formation of lymphocytes takes place in the stem cells present in the bone marrow. These lymphocytes travel in the bloodstream to reach the thymus. These cells differentiate under the influence of thymus and are thus called T lymphocytes. T lymphocytes then re-enter the bloodstream to reach the lymphoid tissues in the lymph nodes, spleen, tonsils, and intestines. Some lymphocytes arising from stem cells in the bone marrow do not enter the thymus. Instead, they go to the other lymphoid tissues. These lymphocytes are called B lymphocytes or B cells. The B in B lymphocytes stands for bursa of Fabricus, a diverticulum of the cloaca in birds. The B lymphocytes in birds are formed here. B lymphocytes are seen in the lymphatic nodules. Like T lymphocytes, B lymphocytes also circulate between lymphoid tissues and bloodstream. B lymphocytes produce antibodies. T lymphocytes are responsible for the cellular immune system, whereas B lymphocytes take care of the humoral immune system. Both require antigens to trigger them into action. Each T cell recognizes a specific antigen, so we have different T cells in the body. T cells respond to antigens by producing clones of T cells. When a specific antigen is encountered in the tissues, the T lymphocytes, which are programmed to recognize that particular antigen, return to their specific domains of the lymphoid tissues. Here, they transform into lymphoblasts. Lymphoblasts then divide mitotically to produce activated T lymphocytes. A small proportion of activated T lymphocytes remain in lymphoid tissues, where they act as memory cells. These cells are capable of mounting a more effective secondary immune system on subsequent exposure to the same antigen again. Humoral immune response is controlled by B lymphocytes. Each competent B lymphocytes are programmed to recognize one particular antigen only. They are released into general circulation, from which they seed the lymphoid tissues, especially lymph nodes and spleen. B lymphocytes don't continuously recirculate through the body, but rather make contact with antigens, taken up and processed by macrophages. Once activated, B lymphocytes transform into plasmablasts, which then divides to form antibody-producing cells called plasma cells. A proportion of plasma cells reverts to B lymphocytes and remain in lymphoid tissue as memory cells. The secretion of antibody molecules by plasma cells takes place either in the lymphoid tissue or at the site of antigenic stimulation. In the first case, antibodies are carried to the appropriate site by both the lymph and blood vascular systems. Clonal selection refers to the splitting of B lymphocytes to plasma cells and memory cells. Antibodies, also called immunoglobulin, are Y-shaped protein produced by B cells to identify and neutralize foreign objects.
An antigen such as a protein has several different sites that can react with different antibodies. Monoclonal antibodies are antibodies that can react with only one particular site of an antigen called an epitope. Consequently, they are highly specific and have applications in diagnostic microbiology and cancer therapy. We can reproduce monoclonal cells by inoculating an animal with an antigen carrying the specific epitope. B cells are then isolated from the spleen, then mixed with a type of cancer cell called myeloma cell that grows continuously. The two types of cells fuse together to form a new cell called hybridoma. The mixture of cells is cultured under conditions which permit growth of the hybridoma cells but not the B cells of myeloma cells. The hybridoma cells are then separated into individual chambers and tested orderly to produce the desired antibody. The hybridoma cells producing the desired monoclonal antibody are then cultured. At last, monoclonal antibodies are then isolated and purified.
T helper cells are another kind of lymphocyte that aided the immune response to an antigen. When a lymphocyte and a T helper cell both recognize the same antigen on an antigen-presenting cell, the T helper lymphocyte responds by producing factors that assist and amplifies the B cell's clonal expansion. In addition, the T helper cells releases other factors that attract B lymphocytes and phagocytic cells. When When a virus or other infectious agent infect a cell, some of the proteins of the infectious agent are broken down into peptide fragments. These fragments are complex with special proteins called class 1 MHC, major compatibility complex proteins, and displayed on the surface of the infected cell. The T cell receptor on the cytotoxic T cell interacts with the virus-infected cell by recognizing both the fragment and class 1 MHC. The cytotoxic T cells then releases toxic substances, which cause death of the infected cell. Cytotoxic T cell also produce a toxic called perforin, which creates holes in the cell membrane, causing the cell to break open.
AIDS. HIV viruses destroy helper T cells. Immune deficiency diseases result from the inability of the immune system to have an effective immune response to infection. Probably, the most common and most devastating immune deficiency disease is acquired immunodeficiency syndrome or AIDS. Two viruses named human immunodeficiency viruses 1 and 2, HIV-1 and HIV-2, caused AIDS by infecting and destroying helper T cells. Though AIDS patients produce HIV antibodies, for unknown reasons they do little to prevent the progress of the infection. One hypothesis is that HIV spends so much of its life cycle inside cells. As the helper T cell population declines, the AIDS patient becomes increasingly susceptible to other diseases. Disorders of the human immune system. There are three types of immune disorders. The first one is autoimmunity. Autoimmunity disorder is characterized by the immune system's inability to differentiate between potentially dangerous cells and healthy ones. Plus, the immune system attacks essential parts of the body. It is either inherited or acquired. There are several symptoms that include painful joints, fever, inflammation, nausea, and fatigue. Autoimmunity diseases also include lupus, psoriasis, narcolepsy, multiple sclerosis, and Graves' disease. Second type of immune disorders is hypersensitivity. Hypersensitivity disorder occurs when immune system reacts to antigens in negative exaggerated manner, causing damage to the body. Hypersensitivity disorder is also categorized into 4 tiers. The first one is a type 1 immediate hypersensitivity, which occurs within minutes of exposure to antigen or allergen. There are several types of type 1 disorders that includes asthma, eczema, hay fever, and food allergies. The second one is type 2 cytotoxic hypersensitivity, which occurs when the antibodies produced by the immune response bind to antigens on the patient's own cell surfaces. This includes Graves' disease, AHA, and Goodpasture syndrome. The next one is type 3 immune complex hypersensitivity. This hypersensitivity disorder is caused by antibodies reacting with antigens, which are then deposited in different areas of the body and can then damage tissues. Last but not the least, type 4 delayed type hypersensitivity. The reaction takes 2 to 3 days to develop. This includes multiple sclerosis, type 1 diabetes, and Crohn's disease. Immunodeficiencies. It occurs when immune response is defective or absent, thus susceptible to frequent and recurrent infections. Immunodeficiencies disorders include Geiger syndrome, selective IgA deficiency, Wiskott-Aldrich syndrome, and Karman variable immunodeficiency. That's all. Blah blah blah. Yeah.